Hello, everybody. Starting our tour of cells, the actual cells. Today's lecture discussion is going to be on prokaryotes. And looking at the chapter 4 that you have, there's just one little section about prokaryotic structure, which is not equal. It's not even close to being equal to the amount of detail we put on eukaryotes. So what I'm recommending that you do is to read 4.3 and then skip on down to chapter 16, part 1 and part 2. And you can go through more of chapter 16 if you like, but basically that's the place where you're going to get a better feel for what's actually there in a bacterial cell, right? Yeah, there's also archaea, because prokaryotic cells, as we learned before, is actually two domains. We've got the domain bacteria and also the domain archaea. The thing is, our archaea are kind of special. In fact, I'll skip down way to the bottom and say that for most of our intents and purposes, archaea are pretty much non-players. If it's prokaryotic and we care about it, the chances are that it's going to be some type of bacterium, right? And by a bacterium, I mean something in the domain bacterium, which is distinct from the domain archaea, right? So they're actually two effectively completely different kinds of cells. They're as different from each other as two cells can be. I mean, they split apart from an ancestor. They did have a common ancestor way back at the origin of all of cellular life 3.5 billion years ago. But they've basically been doing their separate thing ever since, right? And so you can't really lump bacteria and archaea together, except for the fact that they've got the same relatively simple prokaryotic cell structure. But we'll talk about the other kind of cell, the kind of cells that were made out of eukaryotic cells in the next video, right? But the thing about prokaryotic cells, and again, I'm thinking primarily the bacteria, is that they're characteristically simple and small. Simple and small is kind of like the rallying cry, the thing that you need to process and understand and simply accept when it comes to bacteria. And being small in particular, they're taking full advantage of that surface area to volume ratio advantage that we talked about in the previous lecture. Being small, they've got more surface area, more plasma membrane to serve their biomass, and consequently, they're able to do things. Well, like what, Jeff? Well, I'm glad you asked, because it's really most evident when it comes to the potential for growth, right? How does this surface area to volume ratio manifest itself, this advantage? In the form of an advantage in growth rate. Okay, you take a gram of bacterial cells, 1 gram of bacteria. Give it unlimited food, right? And how long does it take to become 2 grams, right? We'd call this an increase in biomass. How long does it take? Well, for E. coli, I mean, this is a really good example, very well studied example. It's about 20 minutes. Okay? In other words, a bacterial culture can double its biomass every 20 minutes. After 40 minutes, it would be 4 grams. After an hour, after 80 minutes, it would be 8 grams, right? And basically, it's increased, this should be 60 minutes. After 80 minutes, it would be a 16 grams. So basically, 1 hour, be able to go from 1 gram to 8 grams. If you were to look at something that's even somewhat larger, I mean, yeast cells are eukaryotes, but they're also very small. So 1 gram of yeast goes into 2 grams. Well, it's still pretty fast, but it's on the order of about 2 hours, right? So what I'm saying here is that in 1 hour of time, a gram of bacteria has increased by a factor of 8. In that 1 hour of time, a yeast culture has not even made it to a doubling point, right? And so, yeah, I think the idea here is that if you're able to convert your food into more biomass, into more of whatever you are, that's going to give you an advantage relative to a competitor that might be wanting to use that same food to turn into its biomass. So again, what is it that keeps cells small? Well, surface area to volume ratio advantages. Okay? What is the actual advantage? Well, those small cells can increase in number. Basically, they're expressing a form of fitness that gives them a huge advantage over slower growing cells. In other words, over larger cells. Okay, so talked about that. Check, check, check. And now we get to the cellular structure of prokaryotes. This is the other part of our discussion. And I'm going to go ahead and switch over to our textbook share. And here we're on chapter 4, section 3, talking about prokaryotic cells, generally smaller, and lists a few of the things that are there inside of a prokaryotic cell. And it gives you a very quick and dirty summary of what you should know about prokaryotes. So quickly, one thing is that, well, I think we'll focus on this one first. This is an important one. The cell wall, right? Now the cell wall, if you can see where it's pointing to this orange layer right here, which is outside of the plasma membrane, the cell membrane, okay? So the plasma membrane is the actual phospholipid bilayer that we've been talking about so far. The cell wall is something that's outside of the cell. It's actually an extracellular structure, right? Extra means outside. And there are actually some other things that are extracellular, like the capsule, like the flagella. But the cell wall, I think a couple of important things is that this is what's going to determine the shape of the cell. Okay. Why is the cell shaped like a little cylinder? Well, it's because the cell wall is shaped like a cylinder. If you took the cell wall away, which is rigid, the cell would lose its shape. It would basically become like a little blob, right? Some cell walls are circular or spherical, and some cell walls are kind of long and spirally. And so the three main shapes that we'll see are rod-shaped bacteria, round bacteria, and we call them spirochetes. We've got these coiled cells that are twisty around, and they cause some pretty interesting diseases. Right? So the cell wall, one important structure. This is also something that is unique to bacteria. The fact that it's made out of material called peptidoglycan. Okay. It's something you'll read about in chapter 16. Peptidoglycan is something that's unique to bacteria. So only bacterial cells have peptidoglycan, which is a really important thing for us to note, because anything that is particular and unique to bacteria is a potential target that we could use to treat bacterial diseases, right? So if you were to administer a drug that would only target the synthesis of peptidoglycan, you would be preventing bacterial cells from making more bacterial cells, because the cell walls would fail and the cells would die, right? You don't have to worry about that same drug disturbing your peptidoglycan, because your cells don't have peptidoglycan at all, right? And so this is a theme that is worthwhile for us to point out right now. And it's one of the features that makes bacteria unique and different from a lot of other things, right? Next thing is that the bacterial chromosome is in a nucleoid region, right? So it's the fact that it's not enclosed by a membrane gives us the fact that these guys have no true nucleus, right? The nucleus in a eukaryotic cell is this thing that is holding all of the genes inside of a cell. It's this little membrane-bound structure in the middle of the cell, right? You don't have that in bacterial cell. The nuclear material, which is the chromosome, the DNA, is just sitting there inside of the cytoplasm in the same area as the rest of the cellular parts, right? So bacterial chromosomes are there. The other thing about a bacterial chromosome is it's circular. It's a big circle of DNA. It's not a long structure like what you find in eukaryotic cells that line up at the metaphase plate. I mean, if you've ever heard about the phases of mitosis, you don't have that happening in bacteria because you don't really even have that linear chromosome structure. Okay. Now, these are all characteristic things that we are going to be coming back to later on. Ribosomes are important. This is where you synthesize proteins. The capsule is an important feature for discussion later on because this is actually something that when you have a capsule, it can make bacteria more likely to cause disease. Right? So what makes a non-pathogenic version of Streptococcus relatively harmless, while a pathogenic one that will cause strep throat, is going to be really dangerous? Well, the one that causes strep throat is able to avoid getting killed by your defenses because it's got a capsule. Okay? Yeah, so the capsule is a chemical coating that comes outside of the bacterial cell. It's like a little a polysaccharide jelly, right? And so these are some of the features that we find in bacterial cells. And you get a brief mention of them here, but we're going to be seeing them again when we scroll down to, get rid of the drawings here, and I'll scroll you down to chapter 16 to see what's going on with that. And so, yeah, I think it's a good idea for you to read about the diversity of prokaryotes generally, because that's really what we should have been talking about in the context of today's discussion. And then when we look at the structures on bacterial cells, you've got some of the things we've talked about, right? The idea that we've got 3 very common shapes in bacteria: cocci, which are spheres, right? Bacilli, which basically means rods, and spirochetes, which are these spirally, coily shaped worm-like bacteria. It's kind of like a worm, except it's shaped like a little corkscrew. Okay? The cell wall of bacteria, a little more detail here. This is where you can see that the material that is there inside of, it's always going to be there in bacterial cell walls, is peptidic glycan. Then it talks a little bit about the bacterial capsule and how the bacterial capsule is something that it causes cells to, one, adhere, stick to cells that it's targeting, stick to other cells in a colony, but it's also going to help protect it from a host's defenses, right? So just want to point something out to you here, and that is that this area here is the capsule, right? That's all capsule. The cell wall thing that we talked about is the cell walls inside here, right? So the cell wall is inside, and the capsule is even outside of the cell wall. The plasma membrane would be inside of the cell wall, right? So there's the plasma membrane. Okay, so both the capsule and the cell wall are extracellular structures, meaning that they come outside of the cells. Okay. Now, in the section of the textbook that we skipped, which is 16.1, I want to give that a quick point out. It talks about the ecology of prokaryotes, and it mentions something that is going to be of some importance to us, and it talks about the microbiome. Right? The microbiome is all of the microbial life, that your microbiota is all of the bacterial and fungal and other kinds of microbial life that's living on you, in you, basically taking advantage of your existence. You're giving all these microbes a place to live. Okay? Now, don't be too alarmed by that, because everybody is carrying tons of bacterial cells. And so the rough estimate that people give is that, well, in the human body, you've got about 10 raised to the 10th cells, right? That's 10 trillion, right? Eukaryotic cells. That's the number of cells that is sitting in your chair as you listen to this video, right? The number of bacterial cells in and on you, well, it's 10 to the 11th, right? So 100 trillion. Bacteria. So you take into account all the bacteria that are living in your gut, feeding on the things that are passing through the gut. Think of all the bacteria that are living inside of your orifices and on your skin. And that, it's probably not a huge mass of cells, okay? But bacterial cells are really small, remember? And so the number of cells that are there is going to be pretty incredibly huge, right? I think I misspoke earlier. The microbiome refers to the sum total of all the DNA that's there inside of your of all those microbes, right? Now, the reason why this is significant to us in 2022 is that every year there are a number of studies that are focusing on the microbiota, right? Your microbiota may be part of what determines whether you are relatively healthy or relatively sickly, right? Whether you are able to maintain a healthy weight, or whether you are you have a tendency towards obesity, right? Whether your immune system is strong, whether your immune system is weak, and obesity. These are a couple of things that are being understood to be influenced by the microorganisms that are living inside of you, right? Which is kind of crazy, right? Because we normally think that we are what our genes cause us to be, or we are things that we control ourselves. We make our own destinies. And now what this seems to be saying is that all those bacteria living inside of us, the huge community of bacteria living inside of us, may be having some role in deciding what kind of person, what kind of life we're living. Okay? And so I think that's it for our discussions from the textbook. I'm going to go back to my whiteboard share and kind of summarize what we've seen so far. So saw the structure of prokaryotes, a cell wall that's made out of peptidoglycan. It's outside of the plasma membrane. Jack talked about that. Bacterial genome is a huge circle of DNA, right? And we sometimes call it the bacterial chromosome, although it doesn't look like a chromosome that's there in our cells. The nuclear material of the DNA is not separated by a membrane from the rest of the cell. We've got proteins for protein synthesis. Ribosomes for protein synthesis. Flagella are there pushing the cells around. I know we didn't really target that, okay? But that's okay, because I don't think there's a reason for us to talk about them later on in the semester. We will, however, talk about capsules as we talk about DNA later on, right? Now, you might think of bacteria as being human pathogens, right? Because that's kind of like the discussion that we normally have when you get into microbiology, which is largely a bacteriology class, study of bacteria, right? You're going to be associating this kind of bacterium with this disease, this kind of bacterium with that disease. And so you kind of get the impression that bacteria are bad things, And yeah, there are plenty of pathogenic bacteria, and we care about them a great deal, right? No question about that, okay? But the vast majority of bacteria are non-pathogenic. I mean, if you just think of the microbiota, all those bacteria that are living on you and in you, outnumbering your own cells 10 to 1, those are obviously not pathogenic, right? If they were, you would be terribly sick, right? These are bacteria that are kind of living off of things that you don't need, right? Your skin is continually replacing itself. And so from underneath, you're always producing new skin cells, and the dead skin cells are out there on the outside, and they're sloughing off, right? You know, maybe some of you are familiar with the idea of what's it called? Exfoliating, right? If you take a loofah gourd and scrub your skin, it's supposed to rejuvenate your skin. What you're really doing there is taking the food away from a lot of the bacteria that would otherwise be eating those skin cells right on top of you, right? So yeah, the cells that are there in your gut are basically consuming the food that you're not absorbing. And so, we have a lot of cells that are in us, on us. They're completely non-pathogenic, and in fact, in a lot of ways, they're helping to protect us from bacterial disease. We know this because in those situations where a person is on antibiotics, which cause the normal microbiota to die back, that makes you susceptible to pathogenic diseases by organisms, yeasts, and bacteria that are resistant to the bacteria. In other words, the reason why those resistant bacteria and fungi are not causing disease in you all the time is because you've got a normal, healthy microbiota occupying all the space, basically taking up all the resources, preventing those bad guys from coming in and invading, right? As you look outside the human body, if you look outside of the body of living organisms, right? Because there are plenty of other organisms besides humans, and each of them are subject to both microbiota and pathogenic kinds of bacteria, right? But if you look outside of all those things that affect animals and plants, most of the bacteria are decomposers. Right? They live in the soil. They basically do for Mother Earth what your microbiota are largely doing for you, getting rid of all things that you don't need, right? And so I wanted to end with that because some people just always think that, oh, bacteria, bad. Bacteria, bad. No. Actually, most bacteria that you come across on a daily basis, all the bacteria that you're inhaling with every breath of air, they're not going to cause disease. Okay? It takes a pretty special bacterium that's equipped with particular features that allow it to attack humans. Those special features are requirements. And so the pathogenic bacteria are always going to be somewhat specialized. And that's a good place for me to end. Next time we'll be talking about eukaryotes.